The most astounding thing that has happened in the last 2 years is one of the gene therapies for one form of the illness. We saw about 40% of people in the trial stop progressing and get better.
There's a lot of promise in applying the leading technology now in the ALS space.
The surviving motor neurons are sprouting and growing, and that's how people are getting return of function.
It warms my heart, and I hope that patients that are suffering today and feel maybe hopeless will be comforted that there's many scientists working every day and clinicians to try to solve the problem, and encourage everyone to just keep feeling hopeful and trying to support and going to the foundations and trying to be part of a community that wants to make a difference.
Now there's even AI challenges globally to try to use all that data scientists have created to try to develop new drug targets and new ideas. So it went from a disease that only a few people were studying to one that there's a global effort to tackle. Can we actually treat before people have symptoms? Can we even move further back like they're doing in Parkinson's and Alzheimer's?
First of all, this is my voice. I'm Tim Green, and I have ALS. But this podcast isn't about ALS. I don't want you to feel sorry for me. I don't feel sorry for me. I'm a father of 5, married for over 35 years, a New York Times best-selling author, an NFL first-round pick, and a practicing attorney. This is Nothing Left Unsaid, where my son Troy and I sit down with extraordinary people for raw, honest conversations about life, its toughest trials, and greatest triumphs. My voice in today's episode is powered by 11 Labs. Today's episode will be a little different. Usually we sit down with one guest, but this time we've got 2 incredible experts in ALS joining us, and I couldn't be more excited about it. Merritt, you've actually been on the show twice before, which officially makes you our most frequent guest, and with good reason. You're arguably the world's leading expert on ALS and one of my closest friends. And Tanya, this is your first time on the podcast. You're doing groundbreaking work at the Institute, developing genetic tools that can target the specific cells involved in ALS, really at the frontier of how we might one day treat the disease at its source. What makes this conversation special is that you're both coming at ALS from different but deeply connected worlds, one from the lab bench, the other from the clinic. And today, we get to explore where those 2 worlds meet. Thank you for joining us today.
Well, thank you so much for having us. Yeah, really happy to be here.
Yeah, thanks for inviting me back.
Could you each give us a state-of-the-field snapshot of ALS right now? What's most exciting, and what do you see as the biggest bottlenecks between discovery and real patient impact? Merritt, you go first.
I'm so excited about just the state of science, the number of people working on ALS and the better understanding of the biology that's happening in the brain and spinal cord of people living with ALS. And so it gives me a real hope that the next group of drugs that are going to go into trials have a much better chance of really making a big impact. I'll give maybe just 2 examples. Last week we had our ALS Trial Network meeting, over 800 people there together, lab-based scientists like Tanya and clinicians, nurses, everybody who cares about ALS and companies. And people are really talking about the ability to subset people by their biology using different tools, like some of them that are developed at the Allen Institute, and really target treatments, to groups of people, a bit like you do in cancer, where based on your biology, you can respond better to certain drugs. And it feels like the ALS field is almost there right now, or we have at least some of the tools. And that makes me really hopeful that we can get better treatments. I can talk more about that later, but that's what gives me the most hope.
Yeah. So, I'm really excited about the new genetic technologies that have come online, that, we're continuing to develop, many people in the field. I think it's for the first time I see the potential since all the work that's been done in the genetic space of applying this technology to genetic forms of ALS. I'm really excited also about learning from the past failures. I think now that we have a new toolkit that we can try to select for the most beneficial outcomes with even the existing therapeutic strategies that have been employed, and to try to tinker with those to, create new versions that may have a better risk therapeutic profile for patients. And so, I think there's a lot of promise in applying the leading technology now in the ALS space. And it's really a very exciting time.
What's one misconception about ALS, whether from the public or even among scientists, that you'd most like to correct? Tanya, can you lead this one?
Yeah, I think that at least I can only speak really to the scientist circles that I run in, because we have, less interaction with clinicians. It's something that we're hoping to change. But certainly in the scientist circles, I think that one of the common misconceptions is that everything is known in terms of the cellular dysfunction in ALS. So we know these cell types that degenerate the motor neurons within the brain and within the spinal cord, and therefore, we can immediately that this is immediately a tractable problem to solve. And so I think the misconception is just that because we have the cell types, we even though there's been failures in the space, that certainly we know enough now. And I think that's partially true, but I also think that there's still a lot of fundamental knowledge that's needed to be able to pursue these various therapeutic strategies. And so, part of this is a balance of the basic research that's needed with the preclinical type development.
Tanya, is the I guess the failures that you're mentioning, is that like genetic attempts in any disease, or does it have to be like, can you learn from other diseases, or does it have to be genetic attempts with ALS specifically?
Yeah, I think I was speaking more broadly, but certainly a lot of the genetic failures come to mind in that answer. I think that, we have, you can look at many different diseases where certain gene therapies or cellular-based approaches have been employed, as well as, small molecules or, ASO-based approach. And certainly there's been, some glimmers of hope. However, there's been some challenges, some profiles that are not ideal. And so I'm thinking very broadly, but also, in the context of just some of the recent challenges in the ALS space.
So I have two. I know you asked for one, but I'll say one misperception is that it's a rare disease. It's not a rare disease. 1 in 300 men will pass away from ALS, and 1 in 400 women. And it gets more common as people get older. And we're already seeing a lot more people with ALS globally. So that's one misconception. The other is that it's not treatable. Now, I'll say that the most astounding thing that has happened in the last 2 years is one of the gene therapies for one form of the illness, SOD1, where we saw about 40% of people in the trial stop progressing and get better. And that told us that it is a treatable illness. I see a lot of patients who come to me and their doctors tell them there's nothing to do, just go home and there's nothing to do. And that's just so not true. So I tried very hard to get rid of that misperception, because there are treatments we can do, and there's trials, and there's a lot of ways to be part of research and part of the solution.
That's obviously how we met too. So it's terrible that it happens, but in that one scenario, I'm glad it happened.
Can I just say something? And this is just a pet peeve of mine, but the trials are only failures if you learn nothing about it. And so that's kind of what I was taught as a young neurologist and doing trials, that you should always learn about the disease, no matter whether the drug works or not. And so the field has actually learned an enormous amount from trials. But Tanya, you're totally right that we can go back now that you and others have developed these amazing tools to see if we might have missed treatments that were effective in different groups of people or different cell types. But I don't actually think there's been that many failures in the field of ALS. There's actually been a lot of successes.
Both of you work at the intersection of hope and hard data. Can you talk about the progress that's happened in ALS over the last 5 years and what you think the next 5 years could look like?
I'm happy to take this one first. First, I'll say there's been huge progress in the last 5 years, and there'll be even more in the next year to 2 years. One is we have we have 5 marketed drugs for people with ALS. So that's huge, including the one gene therapy that has just spectacular results. And the other 4 are, slow down the progression, but they're still important options for people. There's global collaboration on consortium to bring scientists and clinicians and patients together. There's 3 of them, actually, one in Asia, one in Europe, one in the United States. And people are just sharing data, they're sharing ideas, and everyone gets it that you have to move fast in this field. And because of that collaboration, we've seen that most of the genes that cause the familial form have been discovered, about 70 to 80% of them. And that has led to scientists like Tanya and other people coming into the field to understand the biology and develop models and develop targets. And now there's even like AI challenges globally to try to use all that data scientists have created to try to develop new drug targets and new ideas. So it went from like a disease that only a few people were studying to one that there's a global effort to tackle. So the next 5 years, I see us getting more targeted treatments, especially for the genetic forms. I think that the tools are there, and you'll hear more from Tanya about that. But I also think for the people where it doesn't run in the family, we're going to be having more treatment options for people and bigger effects than we've seen in the past.
Mary, do you think there's been enough medicines or drugs that slow it down, that if you're a new company in the space, you don't even target slowing down anymore, you just try to focus on reversal?
It's a good question. And no, not yet. I think that people are still thinking if there's a drug that slows it down 50%, that there's room for those type of advances. Maybe we're not looking for those 10 or 20% anymore. But as the science gets better understood, then the drugs are going to be more likely to be the ones that are stopping it and reversing it. But in the meantime, it's this balance of making a big impact is more reality right now, and the reversals are probably coming in the future, not too far away.
Yeah, so I would say, we're newcomers to the space, as Merritt just said. So I think it's been, we really bring fresh eyes to ALS, and I would say in this last several years, as we've been developing this technology and trying to meet other thought leaders in the field, and meet with various biotech companies that are progressing with various potential treatments. As well as a lot of private foundations. I think that what is a commonality is just so much enthusiasm for solving this, for reversing, coming up with disease modifying treatments. And I think that these global consortiums that are forming, as well as the enthusiasm and the diversity of technologies being thrown at it, really has fostered a very productive, I think, scientific environment that absolutely could give rise to rapid progress in the space, and iterative feedback loops where we're learning more rapidly from the diverse activities that are going on in the community. So I think that, as a newcomer to the space, I really feel invigorated almost daily by some of the interactions I have and some of the messages I feel from people in the community. And, I've worked in other disease spaces before, and, I would have to say that I haven't seen quite this level of enthusiasm. So I think that's really exciting.
Tanya, this is not for Mary, this is for me. What we've seen, my dad and I have seen, is on the kind of fundraising side and on the clinical side, things can be a little bit tribal with ALS. It's one thing we've tried to do is break down those barriers. And frankly, it's one of the reasons we love Merit, is we think she does that, where it's like all about, let's just get a cure. It doesn't matter who does it. Let's just make progress. I probably should say it doesn't matter who does it. And the scientific community, is it competitive or is it more collaborative?
Well, our experience so far has been that the scientific community is quite collaborative. So I feel that many people are reaching out, want to learn more about technology, want to just explore these various spaces without much restriction to it. And so I think, my sense is absolutely it's not as competitive as certainly other areas I've been in. that said, some of our discussions with foundations, I think there's certainly more areas where we feel that, maybe areas have been carved out and foundations are pursuing various, specific initiatives. So it definitely feels a little bit more competitive in the nonprofit space than I would say in the scientific space right now. And, that's something potentially going to be looking to take on a bit with some of the new initiatives that we have going at the ALS Institute next year, where we'll be looking to try to bring more leaders together to try to reduce that competitive landscape overall, because we all have the same endpoint, which is trying to make meaningful, rapid progress.
I was going to ask Tanya. I mean, to have the ALS Institute researching ALS is just phenomenal. I mean, the technologies and brain power you guys bring is unprecedented. So I just was wondering, for the listeners might just want to know a little bit more about the ALS Institute. I don't know if you already have that question for Tanya, but I would love to hear that and what you guys kind of bring to the field when you come into it.
Yeah, well, that's great to hear. We're really excited. Disease research has not been really part of our ecosystem as a foundational of the work for some time. But, essentially the ALS Institute has put in 2 decades of work trying to characterize the fundamental cell types within the brain. And so, trying to understand these, if you say it, like the Legos of the brain, the building blocks, trying to characterize them at the level of, what they look like, how they respond to various stimuli in the brain, who they connect to, what type of basic properties they have, and in the normal or healthy brain. And so, through this kind of classic atlasing type studies where we've been able to characterize and group these cell types at various parts of the brain or the whole brain across various species, we've been able to ask questions about how are these cell types conserved across species, trying to help us to understand which cell types are important in the human and what make us different from other organisms, as well as now trying to better understand the dysfunction of these cell types in various diseases. So it's really, our work is built on 2 decades of foundational knowledge in brain science, and now we're looking to turn that knowledge in towards these various disease spaces. And so, in many ways, a lot of the technology we've developed to atlas the cell types in the brain, we can turn them to these new diseases fairly rapidly with some methodological changes. For example, we focus largely on the brain, but not the spinal cord. So we have a lot of less knowledge and space, and experience with that, but that's something that we think we can grow very rapidly given the foundation that's been built. And that the new initiative that we are looking to launch next year is really the first major initiative at the Institute to tackle disease from this perspective of both the foundational atlasing side of taking patient samples and profiling them, to now trying to think about the therapeutic strategies. And so it's an exciting time. It's also kind of a new frontier for us. The next chapter of the ALS Institute is in this disease and translational biology space.
Tanya, right before Merritt and my dad joined, you and I were on, and you said that you guys kind of got into ALS on accident. Can you tell that story?
Yeah, it was a bit, an accident, I guess. I've been at the ALS Institute now for 13 years, and working on genetic tool development and trying to do, as I said, this foundational biology. The entire time, last 10 years at least, was really just on the brain, and we'd lop off the spinal cord. We wouldn't do much with this tissue at all as a institute. And we had the opportunity to, with a partnership actually we formed that we publicly announced with Biomarkers Pharmaceutical. This is one of the world leaders in gene therapy space. And, they came to us, they work on rare disorders. and developing tools for that. And they have not, until the partnership with us, ventured really into the CNS space. And it was through that partnership that we were able to, work on disease programs that included doing work in the spinal cord and tool development in that space. And I have to say the first tools we developed that hit these spinal motor neurons in the spinal cord, and the first images I saw at the microscope were just spectacular. I had never really appreciated this cell type in the brain and how beautiful it is. And when we first got we got videos of the entire spinal cord, and we're flying through these beautiful spinal cords and looking at the amazing innervation of the spinal cord and to the targets in the body. And, it just really struck me how important, the brain-body interface, I feel like brain-body has only recently been kind of more appreciated, that a lot of neuroscientists who studied the brain in isolation, but not have considered the body. And so I think it was that kind of realization and how we could potentially, do a more holistic approach and focus on diseases that have the center of the spinal cord, that is what kind of led us into the ALS space. But really, it's sometimes just the beauty of the cell types are really, so it, drew me in, I guess. Yeah.
Tanya, I want to go deep on your recent work at the Allen Institute that highlights new genetic tools that can target the cells at the center of ALS. How does that specificity actually work? And why is it such a breakthrough for neuroscience?
Tanya, I'm going to piggyback off that and say, pretend one person on this call, I won't say who, is a lot less intelligent than the other three, and explain it like you're explaining it to him.
No, we're all going to learn.
Yeah, that sounds good. So we've did, the two principal cell types involved in ALS, the motor neurons in the brain and the spinal motor neurons in the spinal cord. We were able to, through a non-invasive, so very easy way, deliver viruses to only target those two cell types, both in the brain and in the spinal cord. And we've been able to engineer these viruses that are not harmful in theory to only express in these regions and not in anywhere else in the body. So not in the heart or in the liver or in sensory neurons within DRGs. And that's really critical because the majority of gene therapies in the market today and that have been pursued use a promoter-based system, or a system of trying to express the gene, how you drive the expression of a particular gene in the brain or spinal cord, in a way that it actually expresses throughout the body, okay? So expresses throughout the liver and the heart. And this is a lot of where the toxicity comes from, at least. That's a leading theory. It could also be just the expression of the viral vectors themselves. But in general, having expression in unintended cells in the brain or the spinal cord can cause a risk profile and, unintended effects. And so, the technology we have really ameliorates or completely eliminates that unintended expression. And we think that's really important for the safety of future gene therapies. And, I think something that else is really important to us is that, there's a lot of progress that's been made in IV capsids or AAVs that you can deliver via the IV route of administration instead of having an invasive brain surgery. And so there's been a lot of progress in that space in the last few years. And so we're really excited about the potential for combining our cell type targeting viral vectors with these IV capsids, the peanut butter and jelly, which would allow patients possibly to have a routine procedure to get these viruses, but not have the risk of it going body-wide and expressing in unintended places. So, that's what we're most excited about right now.
Mara, do you think there's any, obviously, and the genetic version is would help ALS in a whole and help a lot of people, but do you think there are things we can learn? I know you and I have talked about this I don't know, maybe a year ago when we were together in Atlanta. But do you think there's things that Tanya is doing and the whole gene therapy side that can translate over to people with sporadic ALS?
Yes, absolutely. There's actually already a couple gene therapy approaches that are enrolling people without the genetic forms. So they're targeting a protein that people think is important in all forms of ALS. But to be able to do what Tanya just described, to really target it specifically to the motor neurons is really, it is a breakthrough. It's like a huge transformation for the field. Just like what Tanya said, so that you can target where the problem is and you won't get off target side effects. So I'll give you another example. There's there's some companies that are working at ways to get rid of this protein TDP-43 that is aggregated in the motor neurons of people with ALS. It's not in the right place and it's aggregated, and so that happens in 98% of people with ALS. And so they're developing gene therapy approaches to deliver therapies that can get rid of that aggregation or get the TDP-43 back into the right place. And that's for everybody. So you're using the technologies that Tanya described, but it's for the broader group of people with ALS.
And then, yeah, that's awesome. And then, Tanya, you talked about the like doing it through IVs and how much progress that's made. What's the progress on that front? Is it that the IV is more effective, or as they figure out a way to get it to the brain more securely? I guess, what is the progress?
Yeah, so I mean, the main challenge for the field is trying to get something through the blood-brain barrier. That is just a really, really hard, has been a really hard problem, because that's what the blood-brain barrier is designed to do, is to prevent entry of molecules such as viruses. And so, the goal for many companies that have poured considerable, amounts of money to try to find a capsid or a protein coat for the viruses that allows entry into the human brain. And so that's been really hard, but people have made progress recently, and there's maybe a handful of new capsids that have come out that look promising in monkeys. A lot of the last testing is in monkeys to see whether they can get reasonable transduction or widespread expression of a virus when given IV. And so there's several now that look very promising. And the main advantage of this is just the brain surgery that are performed for some of these gene therapies, for example, in the Parkinson's space, are incredibly difficult surgeries that patients have to undergo. So, and these are not scalable, that type of brain surgery. First, these are 13-hour, for example, we want to hit the striatum, a region of the brain that degenerates in Parkinson's, that it's many sites, a 13-hour surgery. It's quite a substantial, only a few places in the world you can get that surgery routinely. So this is not really scalable, any type of brain, a lot of invasive brain surgeries. And so, we want to have technology that is really accessible to all. And so we can think about if we can produce enough virus for our IV route of administration, that is scalable, and that can be done really anywhere. And so these capsids that are coming online, these new proteins that help get across the blood-brain barrier, are really game changers, I feel, for all of these gene therapy, these applications.
When you are developing these tools, do you imagine them as broad platform reagents that can apply to many diseases, or as bespoke solutions built specifically for ALS and related disorders?
We have two major efforts really going on at the Allen Institute. And one is to do large-scale genetic technology and tool development to get comprehensive coverage of all cell types within the brain, and tools that can be used to look at these cell types in various species and to understand these fundamental questions. Then we have, smaller scale work that we have at the Institute, non-pipeline type work that is specifically developing tools with disease indications in mind for translational purposes. And so the tools for the spinal cord are exactly from the smaller subset, this more focused effort to try to make rapid progress in a particular disease that's devastating, that we feel, has some potential footholds for us to get into. And so, we have these two efforts going on because, we want to be able to fuel the community with the tools needed to make these fundamental discoveries that I think are still really needed in every really brain disorder. I feel needs better cellular resolution. And we just haven't been able to do that as a field because the technology has been limited. So, we hope to grow the knowledge on the basic side just as much as we hope that we can do swift boat work and this type of smaller scale focused efforts in ALS and related disorders.
I want to have a little aside, and you could decide whether to keep this in or not. But I know, Tim and Troy, you met Mr. Lee Rizzuto a long time ago, and he was an amazing man who started the ALS Finding a Cure Foundation. But 10 years ago, or 10 or 11 years ago, he told us, the executive committee, go to the Allen Institute and get them working on ALS. And I used to think every idea Lee Rizzuto had was a good one. So he actually sent us on a plane, and we came there. It was, and we met with scientists, amazing scientists, and we had a great 2 days of talking science. And it's great to see it happening. I mean, it was, his idea might have been ahead of its time, but it's just hearing you talk about the way that to deliver treatments to the motor neurons in a targeted, easy, scalable way gets me very excited. I wish he was here to hear it, but I'll tell his family.
Oh, I'm so happy that you came to the Allen. That's wonderful. Yeah.
It's a smaller world, right? I mean, even smaller when you're in ALS, but small regardless.
Merit Your Master platform trial has become a model for efficiency and innovation. For listeners who may not know, how does a platform trial differ from a traditional trial? Why is it so powerful in a disease like ALS? And how can the Allen Institute play a role?
Thank you for that question. Traditionally, when drugs or treatments are developed, you test them one trial at a time, and that you have your sites, you have your participants, and if the drug works, great, you go on to approval. If it doesn't work, you shut down the whole process that you just built up. A platform trial builds the infrastructure once, and it keeps going until you've cured an illness, basically, or developed lots of treatments. It shares the infrastructure between the different companies with their drugs. It shares the data from placebo, and it's super efficient. It cuts down the time to getting effective treatments in half. It cuts the costs in half. And for people who are part of the trial, it really decreases the number of people that are not on the active drug, on the placebo, in the randomized part. So it's an idea that really came from cancer. It's been very successful in breast cancer, lung cancer. Many drugs on the market for those diseases came from platform trials. So we decided to do this in ALS when we saw that there were 200, 300 companies in the space. We wanted to bring that efficiency, and it has been efficient. We tested 7 drugs in the first 4 years. We have 3 more starting, including one that might work on repair. We've had other groups like in Parkinson's and stroke and traumatic brain injury come to us to learn how to do that for their illnesses. So it's the next frontier in brain therapeutics, is to build these platform trials. So I hope we'll work together with Allen Institute as they develop, these therapies now that and that they can bring it into the platform as an efficient way to test them in people. That would be fantastic.
Just add that, we're looking for active, partnerships. And I think, one thing that's very clear in the space that, we will live in the preclinical domain space, and we need the partnership like yours that can help us to translate some of these these potential technologies that we're developing. And so I think, that's the next frontier for us, is trying to identify the correct partners for us to leverage our respective expertise to help us advance more rapidly. So yeah, I would be really happy to discuss, how to do that.
Count me in.
If nothing
That
else,
sounds
if
great.
nothing else comes from this podcast, we got a good match here with MGH and the Allen Institute.
Yeah, that's great.
That's awesome. That would
Thank
be
you.
You know, Mary, you made one comment there. You said we've tested, I don't want to get it wrong, you said 7 drugs in the last 4 years with 3 more starting.
Yeah.
For people who don't know about the platform trial, how long would that have taken ALS before to test 7 drugs?
Oh, that probably would have taken maybe 10, 11 years and cost a lot more. So it's
wow.
super efficient. And of those 7, 2 are going forward to large phase 3 global studies. So we had 2 with some positive results and 5 that were clearly didn't work. But again, we learned a lot from them. The other efficiency I'll say is, when you start a standalone trial, it can take a year from the time you start designing it and you get everything ready. In the platform trial, it's about 30 days. So it's about the ability to get your new idea into to patients quickly. I can't even remember why we used to do it, and probably because we didn't have enough drug candidates or ideas to make a platform trial make sense. But now it definitely makes sense.
I'm obviously very biased because that's what we that's what we raise our money for. It's so good to hear. I know it, and it's still good to hear because it's like, I remember when we first came to see you, it was just Rizual, I always say it wrong, and Radicava had just come out, and now to hear, 7 drugs in 4 years, it's exciting.
Yeah. Season 1 supported helping to launch the platform trial. I was very happy to another disease group came to us and wants to do it in their disease, and they said that the Healy ALS platform trial is considered the gold standard in platform trials. So it's good to hear, but we want to get even more drugs and therapies that work. So we need that partnership Tanya talked about with the labs and the scientists.
And more drugs is just a matter of funding, right? Or no?
It's funding and it's science. I mean, you need both, right? You need the brilliant scientists with the technologies, and then you need funding for their work.
But like right now, if somebody said, "Hey, I'm donating $10 million," right? Isn't it true that instead of starting 3 medicines or drugs, you could start more, or is it not? Is the pipeline not full?
The pipeline is still a lot out there. A lot of companies are struggling to get funds to go into a trial. But 10 million can do a lot of good, both at the lab as well as in patient trials.
All right, Dad, back to you. I'll look under my couch cushion and see if I can find it, Merritt.
Merritt, you've said we're no longer thinking about ALS as untreatable. We're thinking about how to treat it smarter. What does treating smarter look like in practice? Are biomarkers and individualized therapies already changing how you design trials?
Yes, I think being smarter is about having the tools, or again, working with the lab scientists and make the tools so that we can target the right drug to the right person at the right time in their illness. There might be different biologies that happen at the beginning of the illness versus later, so you might need different treatment approaches. And then we really don't think it's all one illness. You might need different treatments. And we finally have, some of the tools, but we need a lot more. We actually launched a new initiative called the Healy ALS My Match program to use the tools we have today to match people to the right drug. So, for example, we can do some imaging studies and measure types of inflammation. And then we can maybe only give people who have that image our anti-inflammatory drugs. Or with genetics, obviously, if you have one type of gene, maybe only those people get the gene therapy. Examples like that to get smarter about how we develop drugs and really help treat different subsets of people with ALS.
Merritt, how close is that to being, I guess, so someone could log on and actually use it?
it's there now, but it's not as good as we need it, put it that way. I'd love to hear Tanya's thoughts, but we have some tools, like we have biomarkers in the blood and the spinal fluid. We can make people's motor neurons in a dish, but we don't have all the ways to tell if, these 5 people have this biology and that drug, and this 5 another one. But it's coming. So I'd love to hear Tanya's thoughts about that, and if the Allen Institute's working on those kind of tools as well.
Yeah, we aren't working on, biomarker assay development at the moment, but we recognize how critically important that is. For example, just trying to pair our selective technology, in the context of ALS. Well, if patients, by the time they're in the clinic and they're diagnosed, have 70% loss of their motor neurons, then of course, the targetable population, maybe it's way too late for selective targeting. So, we see the value in diagnostics much earlier. This is really a hallmark, I think, in many neurodegenerative disorders. I keep thinking about Parkinson's disease, but it's the same case, is that, trying to move the bar so that we can think more about using selective interventions. So it's not something that we're doing at the moment. I think some of the in vitro assay development, like you mentioned, the motor neurons, trying to do more of that type of modeling will help us certainly to further, some of the testing that's necessary before we would want to take those next steps. And so I think it is exciting, some of the advances in that field, being able to do that routinely, the type of quality of data that's coming out from some of those studies, I think is really encouraging. So, I'm closely following these 2 fields and, excited about progress that could be in the near future.
Merritt, if Tanya's precision tools could reliably target motor neurons, what would your first dream trial look like? Who would the patients be, and how would you deliver it?
He's making a match here, Merritt. If you
Wow,
can't
that's a
tell.
good question. That's a hard question. Well, I would love something that is for the majority of people with ALS. So the TDP-43 biology. We haven't really tested a drug that could either keep it in the nucleus or where it supposed to function, or get it back there. And if there was a way to target a treatment that could do that directly into the motor neurons, I think we would be working with you so closely and trying to get that to patients quickly. That's one, but I'm sure there's many others, but I like that one.
Tanya, what about you the other way? What you guys have now? I don't know if there's something on the horizon you're working on, but is there something now that you think, like with the I guess platform, literally, and then kind of upon the platform that Merritt has set up, that what would your dream trial look like?
Yeah, I think it's still really early days for us, but certainly we're thinking about some of the low-hanging fruit targets in the space, and how we can continue to de-risk, or reduce the, I guess, de-risk the technology more. And so certainly some targets like TDP-43, SOD1, that's a genetic population, obviously, but some of these targets have come up, and I think one thing we're excited about is just how versatile the technology we've developed is, in that we can turn on and off the expression in these cell types as well. We can induce the expression when we want to. We can, derive different tools, for example, for gene editing or silencing. These are areas that we're working on now to make the platform as generalizable as possible, so we can take on a lot of different genes, a lot of different proteins, both for the genetic forms and then for tackling, more broadly. And so, I think that I don't have a great answer for the immediate. All I say is that I think there's a lot of potential for the technology to go after various proteins and make some meaningful changes in motor neurons.
How critical is disease stage for intervention? Is there a point of no return in ALS where even the best therapy might come too late?
It's a good question, and I think we don't know. The SOD1 story tells us a little bit. So with that gene therapy that is on the market for people with SOD1, we saw better response if you treated early, but we still saw a response if you treated later in the illness. So that tells us that there are motor neurons that can be saved and they can sprout. But clearly, better earlier is usually even a chance for even more success. And Tanya, if you wanted to add.
Yeah, I would just add that something that has impressed me is that, motor neurons are quite diverse. So some recent studies that have shown that in rodents, but also in humans, that there's different subtypes of motor neurons within the spinal cord, and some of them are more susceptible in the disease states versus others. And we have been able to develop tools to target these subtypes, at least two of the major subtypes. And this will allow us to ask questions such as, what is it about these motor neuron subtypes that make them resilient in the disease? And then, what about the ones that die more rapidly? And what is it about that cellular environment? And, we can then start thinking about, how can we potentially, change the environment to make these cells more resilient or potentially, to the disease. And so I think thinking about also motor neurons as they're not one big class, that they're quite diverse, might help us to think about other strategies that haven't been pursued before.
Last time, Mary, we had you on, I think the SOD1 stuff had just starting, and I think you said it was still too early, or you didn't feel comfortable really talking about the results. Can you talk about that now, or is it still too early?
No, it's all been out. All the news is out, and the drug is on the market. It was really phenomenal. It definitely slows down the illness in almost everybody, but about 25 to 40% of people in the trial, depending on how you measured it, stopped progressing and got better. And it's the first ALS study to ever show that. And it took time. It took 8 to 10 months before we started to see that. And we think it's really repair. It's like as the disease is really pretty much stopped by the gene therapy, the surviving motor neurons are sprouting and growing, and that's how people are getting return of function. There's a whole group of people now talking about ALS rehab, about getting people back. It's just it rejuvenated the field. We always thought it was a treatable disease, but we didn't know until we saw that. So again, that gives hope for all forms.
Yeah, that's exciting.
So it's available in most countries now. There's a few countries it's still coming out in Asia, but most countries it's available.
It's so crazy.
But I will say, like what Tanya said about the ease of administration. So that drug happens to be a infusion in the spinal fluid every month. To be able to give it IV, once every couple months would be, or once would be transformational. So we need these new technologies, even for SOD1 ALS.
Imagine if we brought in some of the world's top AI researchers, say a few from DeepMind, and paired them directly with both of you. Could artificial intelligence meaningfully accelerate ALS research? Could it help model disease progression, uncover hidden drug targets, or even design therapies faster than traditional science? And if that's possible, why isn't it happening already? What's the real bottleneck? Data, collaboration, funding, or something cultural in science?
So, okay, I'm probably the person on this call who knows the least about AI, even though I went to MIT, but it wasn't a big thing back then. First of all, I think it is happening. So part of it was having enough data and good quality data, and we need more, but there is a substantial amount of data out there. So there is this UK initiative. It's a lot of organizations coming together, and they've put out this $10 million AI machine learning challenge to build teams to develop, I think, 10 new drug targets and bring it all the way into trials. And they're forming, it's a global challenge. So people are forming teams right now and getting ready to apply. I'm working with some really smart MIT people to put a team together, but they think that there's enough data out there that you can do that. So I think the main issue, I think, is data, having enough and the right type. I think the collaboration is there. It is, as Tanya mentioned, one of the most collaborative fields, along with the scientists and the clinicians. You wouldn't go into ALS if you didn't really want to help people, and you got to collaborate to do that. So I think it's happening for drug discovery, for subtyping people. I'll pass on to Tanya for her thoughts.
Yeah, I mean, this AI revolution is happening, and I think it's an incredibly exciting time to see where this goes. As Merritt said, there's been major investment across academic, biotech sectors, pharma sectors in AI, and I think the general thinking right now is if we have enough data, large sets of data, that we can do this, what Aviv Regev at Genentech describes as a lab in a loop, that we can iterate, we can make some fundamental kind of predictions, we can quickly test this in a loop, iterate on that, and quickly change the hypothesis or change the direction of the work. And we can do this over and over again to make rapid feedback, to come up with the next generation of small molecules as well as tools in various technological spaces that can advance medicines. And so I think it's a really exciting time, and the Allen Institute has made major investments in the last several years in the AI space. We have a Allen Institute AI initiative, a whole institute devoted to generating and mining these data sets, both in brain research as well as much more broadly. We have incubators that are ongoing through these spaces at various times throughout the year. And in brain science, we are actively working to try to make sense and derive more meaning out of these large-scale data sets that we've generated over 2 decades. And so we have a brain knowledge platform effort that's going on now. We have all of the access to the data. It's open access to anyone in the world can come to our website, can learn more about their cell types of interest within the brain, and do specific queries on that through the online tools to, for example, ask a basic question like what proteins are expressed in this given cell type or in motor neurons, or what types of, as I'm describing in my last answer about the diversity of motor neurons, both in the brain as well as in the spinal cord. You can actually see that by going to our website and doing queries. But, we know that right now we're just scratching the surface of what we can derive, the true biological insight, without the additional computational power and the AI can bring to it. So we're really excited. We're pouring a lot of, I think, dollars into this effort, as everyone is in the world. And I think it's surely to bring some really exciting and meaningful progress in the space.
Is the thought that the AI can just basically synthesize so much data so fast that you can almost like give it the like what a drug would achieve? Like if a drug achieved X, how would they react? And it can just predict at a higher rate, a higher accuracy rate, I guess I'll say, because there's so much data it can synthesize. Is that really what it boils down to?
Yeah, it can derive new insights, that with that scale of data, as well as make predictions that without the data being there. I think that's one of the important things, distinction as well too, that if you can have enough data to create these models that can be highly predictive of things, then actually the backwards logic that you need to fill in the gaps is not as necessary. And so I think that it depends though on the quality of the data that you have, and the scale of that, of course. But yeah, that's essentially the idea.
Where do each of you see the next big pivot coming in ALS research? Gene therapy, small molecules, stem cells, AI-driven discovery, or maybe something we're not even talking about yet?
I think this tough question, Tanya gets to answer first. So,
Yeah.
it's a great question.
It is a tough question. I mean, I think I follow the stem cell therapy and gene therapy spaces the closest. I think I've been so impressed in recent years, and I follow a lot of the work in the Parkinson's disease space as well. We've been working in that for a number of years. And, I'm really excited about potential for some of the cell therapies. Certainly, there's a lot to learn in terms of the durability and efficacy and how those can be, what are the primary endpoints that are being measured in some of these work. But I think I'm really excited about that field in general. For the gene therapy space, of course, we're quite biased, so we think that this is really a new frontier in cell types and in the fundamental biology of the brain and the spinal cord. And we're excited about selective precision therapies, and also trying um, the idea of more personalized medicines that in the gene therapy space. And by that, I mean, tinkering at levels that can help us to have more precision overall in the body. And so I think, those are the areas I'm most excited about right now. And I think there's the most room for progress that could have a meaningful impact rapidly.
Yeah, I agree. I think it's exactly what you said, the precision therapy to be able to tailor treatments better for patients. And those treatments might be gene therapies. They might be something we haven't thought about, or I know there's a lot of interest in these nanoparticles to deliver treatments to the brain. So there's all sorts of new things coming about how do you get drugs and therapies in the brain, including nasal sprays. So, but I think it is going to be about this targeted approach. There might always be some drugs that are going to work on everybody, but it might be a combination of approaches. The one area I haven't seen too much in ALS is on neuromodulation. This is the idea of stimulating the brain or spinal cord, either with electricity, magnetic fields, or sound. There's a lot of that in Alzheimer's and Parkinson's and psychiatric illnesses. And it might be that it's not going to work in ALS, but that might be one of those areas where we'll be surprised in a couple of years to see that also as an augmentative approach. And the last area I think is we're starting to move a little earlier. Like, can we actually treat before people have symptoms? As we want to keep developing treatments for people with symptoms, but can we even move further back, like they're doing in Parkinson's and Alzheimer's? So hopefully, and there's a lot of big data studies ongoing to try to be able to do that as well.
What's the thought with that, Merritt? Is if you could detect it, I mean, I guess you'd have to go and get tested somehow. If you could make reliable tests that were cheap enough for people to do at home, and if they could test that they had it, they could start, whether it's Radicava or whatever new medicines are coming out before they even have it.
Yeah,
Well,
kind of
before
like
they
you get your colonoscopy when you're 50, or your mammograms. I mean, it's for a rare disease you have to have a very good test so you don't have a lot of false positives. But I think there's now global studies to follow people at high risk for ALS or gene carriers before they get symptomatic, to try to understand their biology before, with the thought that what you discovered there might actually be applicable to other people without the genetic forms. So I think we'll see a lot of those results out in the next 5 years as well.
Finally, if you could say one thing directly to patients or caregivers about what's really happening in ALS research right now, something the headlines missed, what would that message be?
You guys are both too polite.
No, we're stalling to think about it, but I'm happy to go first. I think a headline would be that there are things that you can do today to slow down your illness, but there's also ways to be part of the solution, and hopefully that part of the solution will help you as well. But to really go to a center where they're doing research, be part of research, be part of this amazing community of patients, scientists, clinicians who are working together to solve this. It'll keep you up, and in spirit wise, and it's how we're going to really get the treatments in this illness. So have hope and be part of the solution with the whole team.
Yeah, I would echo a really hopeful sentiment. I think that all of science, neuroscience and, translational neuroscience is moving towards a big team science collaborative approach, and that will drive rapid progress. We're sure of that, and because there's many evidence for other fields where people coming together, bringing diverse expertise, they can really, really accomplish very meaningful things. And so, I think that's a real source of encouragement to me, that there's an enthusiasm for working together, and that paired with the technology that's changing, and AI and other things that are coming online, I think that we're going to see we're living through a really revolutionary time in science. And I feel so privileged actually to be a molecular neuroscientist during this time. It's really, uh, um, it warms my heart, and I hope that patients that are suffering today and feel maybe hopeless will be comforted that there's many scientists working every day and clinicians to try to solve the problem, and encourage everyone to just keep feeling hopeful and trying to support and going to the foundations and trying to be part of a community that wants to make a difference.
And then before we wrap up here, everyone knows, like we say in every episode of the Merits on or not on, if people want to support what the Healy Center is doing, go to their website, go to tackleals.com. We're raising money, and it all goes directly to ALS Research. Tanya, what can people do if someone listens to this and they're inspired and they say, you know what Tanya said, something just clicked in my brain? What should people do? Should they go on your website? Should they go on, should they call in? So what should people do?
Yeah, please reach out directly via our website. We have an excellent team here at the Allen Institute that is able to to filter questions that come from the community to the appropriate scientists. If you're a scientist listening, you need help to have the tools to make some questions and try to advance your cause as well. We have teams that will help us with that communication and getting you the materials that you need. I think, we're here to help and here to really engage with the community. Our mission at the Allen Institute is to make a difference in humanity. That's one of the foundation principles. And so, we're open. We're called Open for Science. That's our logo. So we're here and supportive.
And then the real last thing before we wrap up, I normally say it 6 times, Tanya, but the real last thing before we wrap up is we have to do a follow-up episode in I don't know how many months. I'm not going to give you guys too long of a deadline. After you guys collaborate and we figure out some way that you're working together, we'll have to do a follow-up and talk about it.
Sounds good. I like that.
Yeah.
Maybe we'll do it with Tim on 60 Minutes. I think I'm waiting for the third 60 Minutes
That's right.
as well. Tim's been on 60 Minutes a lot.
Oh, wow. I have to check that out.
But yes, I love that you introduced us. I think the first person who sent me your paper was Troy, but then I got it from many other places. It's really a phenomenal paper. So.
Oh, thank you so much. Yeah.
Merritt, Tanya, thank you both so much for joining us today. Every time I have these conversations, I'm reminded that progress in ALS doesn't happen in one place. It happens when the lab, the clinic, and the patient community all pull together. Tanya, the work you're doing to build the tools that let us actually see and target the disease at the cellular level gives people like me hope that we're finally learning how to fight smarter. And Merritt, you've been leading the charge on getting those discoveries to patients faster, not someday, but now. To both of you, thank you for the important work you are doing and for sharing it so clearly today.
Thank you, Tim.
Yeah, thank you so much for having me.
If you enjoyed today's episode, consider supporting the podcast for free by subscribing on your favorite podcast platform, leaving a review, and sharing it with your friends. Your support means the world to us. This episode of Nothing Left Unsaid is brought to you by 11 Labs. Their groundbreaking technology makes a huge difference, and we're grateful for their support. To learn more, visit 11labs.io. A substantial portion of our sponsorship proceeds go to tackleals.com, funding cutting-edge ALS research. If you'd like to join the fight and contribute, visit tackleals.com. Thank you for listening, and we'll see you next time on Nothing Left Unsaid.